What is the scientific basis of Ashkenazi Jewish genetic diseases? Imagine a community with a history that shaped its genes in a very specific way. That's what makes Ashkenazi Jewish genetic diseases so interesting and important to understand. These diseases happen because of unique population factors that affected their gene pool over centuries. A key reason is the founder effect. A small group of ancestors started the community many centuries ago. When a small group starts a population, certain gene mutations can become more common than in larger populations. These mutations then get passed down through generations. Over time, this leads to higher chances of inheriting specific genetic diseases. Another factor is population bottlenecks. These are events where the community's numbers drop suddenly due to hardships like persecution or disease. When this happens, only a few individuals' genes survive and are passed on. This reduces genetic diversity and increases the chance that everyone carries the same mutations. Most Ashkenazi Jewish genetic diseases are inherited in an autosomal recessive way. This means a person needs two copies of a mutated gene to develop the disease. If they have only one copy, they are a carrier and usually show no symptoms. But they can pass the mutation to their children. If both parents are carriers, there is a 1 in 4 chance their child will have the disease. Certain mutations are more common in Ashkenazi Jews. Diseases like Tay-Sachs, Gaucher, familial dysautonomia, Bloom syndrome, and some types of cystic fibrosis are more frequent. For example, Gaucher disease results from mutations in the GBA1 gene, which affects how cells handle certain fats. Tay-Sachs is caused by mutations that affect enzymes responsible for breaking down toxic substances in nerve cells. Genetic studies show that Ashkenazi Jews have a relatively uniform genetic makeup, with specific patterns that set them apart from neighboring groups. This makes it easier for scientists to identify disease-causing mutations. It also explains why these mutations are more common in this community. Most of these mutations are not the result of natural selection. Instead, they are due to chance events in history, like population bottlenecks and genetic drift. Some early ideas suggested that carriers might have had some advantage, but current research favors the idea that chance played the biggest role. Understanding this scientific background helps in developing screening programs. These programs test for carrier status before or during pregnancy. By identifying carriers early, couples can make informed choices to prevent passing on these diseases. This approach has successfully reduced the incidence of diseases like Tay-Sachs in the community. Genetic counseling and testing are vital tools. They help individuals understand their risks and options. Knowing the genetic basis of these diseases allows healthcare providers to offer personalized advice, early diagnosis, and treatment. In essence, the higher prevalence of certain genetic diseases in Ashkenazi Jews stems from their population history. Founder effects, population bottlenecks, and recessive inheritance all play a role. Thanks to ongoing research and community screening efforts, many of these risks can be managed effectively.